Thank you, Emily. Welcome everyone this evening. We thank the Nick and Manu Foundation for making this wonderful lecture series possible for the past 30 years. We, the scientists in this crowd, the posters that you saw on the hallway, work in a field called active matter, which basically boils down to figuring out the physics of living systems. Newton's third law, whether it's a bus breaking on a street or a cell breaking at the point where the wound needs to heal. So how do the laws of physics allow for all of the phenomenology that biology has is the game that we are playing. We're a collection of physicists and engineers addressing problems associated with living systems. How is fluid flow accomplished in me, through my blood, or in the plant, in water, or whatever, and how is cell division happening? And you seem to be violating physics by making babies, but it should be physical. So okay. And we are physicists. We live on the charity of society because most of the things we do is not economically viable right now. And we are super grateful for enabling us to do this, especially in this awesome environment where all of the mountains for us flatlanders give opportunity for big things and opportunity to talk to our colleagues. So we're really grateful to the Aspen community for allowing us to do this. Now, our speaker today, Manu Prakash, is the exception that proves the rule. His utility to society is like completely defined within his life. He's not living on anybody's charity right now. So how does this come about? So first I want to paint a picture of Manu as a physicist as I make out based on reading papers and things like that. I think Manu loves flows. He loves flows generated by microorganisms, and he loves microorganisms organized by flow. And this mathematical realm, he has explored with many different tools and many different lectures and given the flavor and some of the things that he will tell us. But through his proof and science initiatives, he has made possible very, very low expense devices to be available to people across the world for everything from science is best performed collectively. Any given scientist is stupid, but the integration of our knowledge of all of the scientists gets humanity forward. So let's increase the number of motivated scientists by handing out simple scientific things to people that they can play with and get their scientific curiosity going. During the pandemic, there were a number of initiatives from his lab associated with things like a diagnostic masks. That pseudo-mask was great. It was a snorkeling mask with some gadgetry around it so that it looks like it functions as an N95. And open ventilators, things that you can find on the web and manufacture in your local country, use it for these purposes. And beyond doing all of this, all of the fabulous science and all of these fabulous initiatives that benefit society right there, he's a diligent member of the community. He does more service to us by teaching us at conferences and the workshops and things like that. I can't imagine how he manages to do all of that. So I'm a fangirl and I'm looking forward to making that.
Thank you so much. Thank you all for coming in the evening. I know lots of people have lots of things to do in evenings, but this is really a delight and personally a humbling experience. I never finish my talks on time. So I want to start with acknowledgements before I dive into what I want to talk about. Everything that I'll tell you today has been a journey for the last, oh yeah, I can take the mask off. For the last 10 years or so, and there is both rowdy, wild, crazy sets of individuals that have come along on this journey with me, and I'm really grateful to have such partnerships. Many of the current members are listed here, many of the members that have gone on, take some of the philosophical principles that we abide by out into the world itself, and this is really a collective journey itself. So I want to talk about curiosity. And I want to talk about curiosity in the context of life sciences. And I'm going to share this view, sense that I often have in terms of thinking about how life sciences has shifted over the last many decades, where sometimes in trying to cure and find urgent solutions to urgent problems, we end up putting curiosity on the back burner. When you think about ecological challenges that we face, we often realize we're trying to fix systems that we don't actually understand. And in the light of that, I want to put curiosity front and center in the context of what I titled this talk as recreational biology. So this is really, you can doodle, you can think with me. The purpose of this talk is to share with you some puzzles that I've been thinking about. But the notion of recreational biology is very deeply connected to, of course, an idea that you might have all heard about, which is recreational mathematics. This is a fairly old and established art in some sense. And in the context of a definition, the notion is sets of problems that you can share with anyone in no jargon, as simple a language as possible to get them excited about ideas. Some of those ideas might be very deep in the context of the mathematical realm, for example. And all of you have played tons of games and boards or, you know, played around with the Fibonacci series or the original story of how we went on to get to graph theory by trying to solve some puzzle of the 7 bridges. One of the things that it reminds me whenever I encounter the problems in recreational mathematics is that the joy is at the front and center of the process. I think as scientists, and many of the scientists that I can recognize in the room, this is something that is true. But do we actually communicate that? Do people who perceive and think about how we do science actually realize that we are all having fun? Although we're very serious people, sometimes that gets lost. So I want to get back to making sure that we spend that time. In the context of recreational mathematics, there have been champions along the years. And you know, how many of you have read work from Martin Gardner? Just curious. Okay, so that's 50% of this room. And if there is one thing you take away from this talk, go read one thing written by him. It would delight you in ways. He was not a trained mathematician, but many things like the game, Conway's Game of Life, Penrose tiling, were all popularized by his writings. And this is really in the 20th century. He wrote hundreds of books on recreational mathematics. There is no topic in mathematics that he did not touch. You know, you go back, of course, many of you have read Lewis Carroll's Alice in Wonderland, and he was also a mathematician and spent a lot of time creating and inventing puzzles. And you know, one of the favorite ones of mine is a book that got published after he passed away. You could go further down, all the way in the 9th century, you can find Latin literature that's really focused only on recreational mathematics. And the oldest example, when you start thinking about, is much more in the Chinese mathematics literature of the magic squares, which is called the Lu Shu. The way you think about this is really the classic 3x3 magic square. Even numbers are represented by white dots, odd numbers by black dots, and you can think in terms of organizing this into a magic square. Now, one of the things that, if you are to translate some of these ideas to biology, you might wonder, actually, yeah, before I jump into, if you get completely bored in this talk, here is your recreational mathematics puzzle. It is possible that the puzzle I'm about to give you is connected to the content of this talk, but it doesn't have to. You have a doodle or a pencil or a pen, you can try doodling it. You could also try to do this in your head, which is you are given a donut and you must divide it into maximum number of pieces with 3 straight planes. Okay, so is that clear? Something like that. And I'll let you think about this. Anybody who gets the answer, just shout out in the middle of the talk. I am actually curious how many of you will do it. So now going back, is it possible to take ideas from recreational mathematics and the philosophical framework and apply that to biology? In mathematics, we have the number line. We have remarkable sets of objects that we handle and play with. And you know, you can naturally start thinking about many of these sets of puzzles arriving. Oh, I'll do that in a second. Thank you. But ironically, you know, biology has that too. That's the biodiversity on our planet. Anywhere you look, we are surrounded by life. This is literally millions of species all around us that teach us rules of biology. And without comparing, it's sometimes hard to even think about what you might learn in one system is how applicable that is to a broader group. I love this. This is one of my favorite illustrations. It's a graphic illustrator, Charlie Harper. What I love about this representation of biodiversity on the planet is that the lion in Giardia, which is a microorganism, and lion, which many of you might have met or not met, are drawn at equal scale. And one of the contexts that I think about in terms of our knowledge of life science on the planet, you can learn from every one of these organisms. It's often said that for every single problem, it might actually be the right organism to ask that question. Mm-hmm. Of course, this is not just randomly distributed. It's because we have all the strong evidence to believe that there is a single origin of life. Much of this is organized around in a tree, and hence there are certain sets of rules that you can compare any of the findings that you would explore in this tree of life. And despite of this incredible resource that we have as life scientists or people that deeply care about life, with the estimated 9 to 10 million species on our planet, majority of what we know in detail from a life science perspective comes from around 10 species. Just think about this for a second. If you are an astronomer with the ever-expanding universe right next to you, and you decide to learn everything about astronomy from 10 objects, this has been something that both life scientists, ecologists have been struggling and fighting over, and this is just the right time because the kind of molecular and cellular and imaging instrumentation tools have become available that you can literally pick any corner of this tree of life and make discoveries to really start teasing apart and building what we would all love to discover in terms of rules of life. You know, ironically, I'm going to give you one example of recreational biology for you to believe me that sometimes tinkering and wandering actually does lead to some valuable insights. So you can see the sequence. The sequence was published in EcoLive, and the genome was published 1987. Somebody named Yoshizumi Ishinua noticed something strange about that sequence. So I'll give you 30 seconds. You'll find something interesting. If you were sitting, you know, at that time, you couldn't just sequence so easily. You work very hard to sequence. You would get this string of letters. Ishinu noticed something in that string of letters, and I wonder if you can see it. It's a little bit hidden, but it's there. And it's a very simple mathematical idea. Think about symmetries. You find, if you were to split this, does anybody see a palindrome in there? So right there. So in the context of DNA sequences, A pairs with T, G pairs with C, A, T, G, C, you can see that around that, the two arrows, there is a palindrome. This is something somebody could have looked at and said, okay, you know, no big deal, I'll just move on. What does it mean? He thought about it, and he actually published that work discussing this framework. Not only did he notice that, he noticed the fact that these palindromes were arriving and appearing with very specific spacer gaps, constant spacers. So you have a little palindrome right there in the pink spacer, another palindrome, another spacer. And that's where 1987, he published a paper. Nobody cared about it. What does it mean? Until 1993, someone now studying archaea, completely different domain of life, finds exactly the same pattern in essentially another sets of sequences. Same idea, palindrome, another spacer sequence, palindrome. And because of the fact that in completely two domains of life, this idea was persistent, everybody got very excited. You know, when you think about a generic framework that applies to a broader tree of life, people start thinking in terms of its value. That led to, of course, massive number of sequences being published. And in the 2000s, people discovered that these spacer sequences actually match exactly the viruses and the kinds of pathogens that are attacking these bacteria in archaea. And most of you probably know this and have realized by now that is what CRISPR is. It is one of our most powerful tools in biology, but none of that would have been actually possible until at that time, if somebody actually takes that time to explore and just share what they explore with no sense of goals sometimes. There was no goal associated with that at that time. So in the context of this type of an approach, I think Niels Bohr, a letter to Niels Bohr, which Max Delbruck wrote, really sums it up in terms of the spirit of this. I was quite depressed around this time because the structure of DNA had been found, and a lot of the molecular puzzles he was excited about had actually been discovered. And he wrote to Niels Bohr when they were discussing what he's excited about, that he will run into paradoxical situations analogous to that which classical physics ran in its attempt to analyze the atomic phenomena. This, of course, has been my ulterior motive in biology from the very beginning. What he was talking about, taking a new approach to science in his second stage of his career. You could read some of the work from that stage is dramatically different from what he did in the first stage, which is about paradoxes and really embracing the complexity of life on our planet. I want to take you on a journey on a few organisms. It's going to be swampy. We will be in mudflats in the ocean. I am going to pass through, I have 200 slides, so I'm going to pass through many sets of organisms. My goal is to not explain to you how these things work, but share what keeps me up at night and the kinds of puzzles that if you just sit down and think about, you can actually crack and solve yourself. So let's go. I think one of the things that's very clear, there are many biologists that operate and work in the organismic scale where they're really trying to think about the whole organism. There are many other biologists that purely work on molecules in a molecular context. We often think about it is necessary to actually bridge these gaps, and individual cells and cellular scales are a very important bridge. We'll focus quite a lot on a cellular scale, although there is a lot of recreational biology to be done. Most recently, shape of an apple. You can stare at on a leaf for hours and hours and discover incredible mathematical puzzles, which many people have done. If you're interested in playing with molecules, which are much harder to play, much more expensive, you don't see them. A colleague of mine developed several class of online gaming tools. This is one of the most popular. It's called Eterna, where you can fold RNA in all kinds of ways and forms. And one of these sets of tools have been actually used for an incredible number of contexts and challenges in medicine itself. But we're going to focus on this kind of a missing scale of the cellular side of the story today. So for some of you, this is what you were told that a cell is, and you were lied. I apologize. This is what a cell is. This is literally only a handful of the diverse geometries and morphologies that you might find in eukaryotic cells. And often enough, when I'm thinking about this in terms of what does it all mean? What do these geometries actually do? And I'll give you several examples today where we have absolutely no idea what these geometries do. And of course, this is a significant portion of the eukaryotic tree of life. This is not just a small portion. It turns out as animals, we pay a lot of attention to animals itself. Although if you were to think about the eukaryotic tree, almost out of the 16, 13 of those same phyla decided to remain unicellular. This is a way of life. This is not a lower form of any kind. It's just they are as evolved as us, except we understand them very little. Okay, so the other aspect which Aparna alluded to as well, cells are not just stationary. I'm assuming most of you have seen that crawling cells video before. But you know cells crawl. Actually turns out cells walk. This is a euplodes, a beautiful system in which microtubule machinery connected to these joint cilia enable a walking gait in a single cell. Cells hunt. That's an example of a cell that we may get a chance to talk about today. That is not a worm. It's an actual single cell trying to find its next prey. So one of the things that we will spend some time on, let's start with puzzle number one. How would information processing work in systems that don't have a brain? Most often we are told in terms of the history of neuroscience that you start with a unit called the neuron, you add that up and you can get complex behaviors. But I can tell you, you can spend hours looking at that hunting cell video and you could not decipher it from the kinds of behavior a worm might be exhibiting. Where would that processing come from when you have a single cell? Again, this is all real-time behavior, so you can't do much of the transcription translation, which are long-term behaviors that you could suggest. These sets of behaviors have to be encoded somehow in real-time phenomena, which is where active matter would come into play. of the best books in this context, other than I absolutely don't like the title, was from Jennings in 1906, Behavior of the Lower Organisms. What I don't like is lower. What does that mean, lower organisms? And essentially, Jennings was told to sit down, and much of his work was discarded because people at that time had such a strong belief that single cells and single organisms could not encode complex behaviors. That has been now refuted. Jennings' work has now been redone. Fortunately, very recently, I talked to Jeremy, who did the work at Harvard. The cell that Jennings found and did the work, they were able to find that cell, they were able to do the work, and then the cells were lost. You can't find the cell again. So, you know, this is what it, when you're going out there, this is the harsh reality. So little that we can actually recapitulate in terms of even getting into the lab itself. So one of the things that we will talk a little bit about is certain sets of ideas around how would you encode sensory motor systems in active matter. And in the simplest possible form, this is a classic framework. We call these perceptive machines. The idea that there is an environment, which is the world, there is some sensory input, there is an internal state to the system. That internal state changes as a function of a sensory input. The action can both change the environment and can change the internal state. And we will think about this framework in several different sets of systems. So let's start with the first one. You've all seen this movie before, but just as a reminder, that's a neutrophil crawling around, trying to catch a bacteria. This is probably the most watched video in cell biology. Unfortunately, most people have never seen Jennings himself. And one of the things that I love about what was described in the context of this is, I'll just read this. Anybody who's watched now a famous movie made by the late David Rogers in the 1950s of a neutrophil chasing its bacterial prey amongst the red blood cells cannot fail to have been impressed by the ruthless efficiency of the predator, despite its apparent senseless simplicity. And I love that phrasing because, you know, it's trying to allude towards that there must be some simplicity in this framework of that chase. And in the lab, almost 7-8 years ago, a graduate student, Nate Sierra, another postdoc, Adrian, we started thinking about what would be the simplest chemotaxis system that you could make. And of course, the simplest place to start is abiotic. We actually accidentally stumbled upon a system that I now just let you appreciate. This is a system that you can create in your own kitchen. Go find some food coloring. You can buy it at a grocery store. Find a clean glass slide, which I have right here. If you can't find a clean glass slide, you can take a lighter and burn the fingerprints on a glass slide, and then you're ready to go. Put some droplets, and this is what you're going to see. What you're watching is almost real-time videos. I think this has been sped maybe 2 or 3 times. Different colors starting to both sense each other. They are surrounded. They have a motor inside. Clearly, this is not a perpetual motion machine. Turns out there is a tornado inside them. You can visualize the tornado. When the tornado destabilizes, you essentially get motility. But the fact is that they can sense each other. They are talking to each other at long distances, very much in the same framework as you would think about how a cell would do. They also have a sense of self. They can detect each other. And again, you know, I think rather than telling you, much of this is published, of how something like this works, what I would love for you to do is to actually try it. It's one thing for me to tell you how this works. It's another for you to actually play with this. In the spirit of recreational biology, I'm telling you this is the simplest chemotaxis system that has been ever built, except I'm not going to tell you how it works. Something really remarkable happens in systems when you think deeply about them. One of the things is not only do these sets of droplets know each other, they are talking to each other, there must be some mode of communication. They can talk to themselves. The fact is, if there is a wall, they can see their reflection. And ironically, when we discovered this, the idea of a self-generated gradient in 2017, 2016, in the paper, we ended up including this line. We're not aware of any natural example where the direction of a boundary can be determined by a diffusive sensing, but hypothesize the existence of this mechanism in biology. you know, editors may or may not like it. We just post papers on archive, so we can write whatever the hell we want. Ironically, 2020, we did not have to wait. 3 years later, Robert Insull's lab published this paper where they actually demonstrated that single cells have the capacity to generate their own gradient, and the exact idea of looking around the corner is implemented. And of course, because they are British scientists, they had to implement one of the mazes that's known in the palace from 300 years ago. Humans get lost in that maze. Cells have a pretty good time navigating this maze, finding really the optimal paths rapidly. So, you know, this is what happens sometimes when you think about just a set of an idea that you share, and eventually that leads to different sets of people being able to take that in the context or the problems that they might actually care about. We've been playing around a lot with building collectives of these. Again, very simple idea, just with a Sharpie, a ruler. You can start now playing with collective droplets. I'm not going to spend too much time on, other than the fact that make beautiful, these pictures make physicists happy. Oh, let's go back to geometry. Let's think a little bit about what geometry might have a role to play. I'm going to check if I have an echo now. So let's focus on one cell. This is one of my favorite cells. I have many favorites, but this one currently is my favorite. This is that cell that I told you about that hunts. This is Lacrimaria oler, an incredible cell in terms of its behavior. I'm going to show you tell you the little story, the backstory. These are the sets of people that have been involved. Scott Coyle, who was a postdoc at that time, joined my lab, and we have a tradition in the lab to go to the swamp, put rubber boots. I have not lost a graduate student or a postdoc yet at the swamp, but this is one of the swamp and marshes we go. I'd been searching for this cell for the last 7, 8 years, and we get out there the first time we test the swamp. It's filled with Lacrimaria. I got delighted. We brought the cell to the lab, we cultured it, we discovered something quite remarkable. That video, or the first footage of when we discovered the cell, was actually taken with a foldscope. And I'm going to show you a recreation of what we saw in the field during that time. The only thing that's not true in this video is the cells are not creating the music. Music has been added. So here is a single cell. This is real time. I can watch this for hours. You can see this is literally a 5-minute video. What is this cell trying to do? What is it thinking? You will see intruders passing by. These are essentially other ciliates, and it's a hunter. And watch right there, what happens? That's a strike. Poor cell gets ingested in real time, and in a few seconds, the cell is not still, hasn't filled its belly. And to anthropomorphize it a little bit, I'll call the extruded part neck. The tip of that we will call a head. The body we will call its body. There is no neck, head. This is a single cell. Just for terminology, we'll use that. Again, let's just watch that. Another instance. Right there, there's a strike, ingestion, and the process continues. Now, what's really surprising, I didn't tell you the length scale in the problem. The object that we are looking at is only around 50 micron. The cell that you're about to see, it's another in the wild type, it's just right here, 50 microns. You have a protrusion that is almost a millimeter long. And I'll let you play this. Watch right there. This is still the same cell. And that's the strike. It's going to reel itself in, and over time, ingest this entire organism that is almost actually the same size as the cell itself. So we wonder, how would something like this occur? How would you search space? How would you encode search algorithms in a system like this? There's a lot of things going on that I don't have the time to talk about. We often think about problems in the context of the extremes. The simplest way to think about this is the largest strain and strain rate that has ever been observed in a single cell, because the videos that you were watching were all real time. And one of the things that we can do is we can grow them in the lab, we can create cultures, we can observe them, millions of data points, incredible amount of behavioral analysis that we can do on this. They do not eat each other, by the way, if you were just wondering. They do know a sense of self. I've never seen cannibalism in these systems. They also don't knot themselves. But let's just look at it a little bit closer. You'll notice a very classical thing that many of you mechanicians in the room might notice. There's a little bit of this buckling dynamics that you actually saw. Of course, if I was to translate all of this behavior in the context of frame of reference of the cell, you would see a kind of a search cloud being searched. And then, yes, I have 5 minutes left. Wow, okay. What else?
Okay, let's go for 10 minutes.
Yeah, let's, I want to give you maybe I'll give you flavors of puzzles. And okay, you saw a higher resolution version of that feeding, and you can see that the organism is essentially morphing around to try to eat something. Now, how would something like this work? And instead of telling you, I'll just give you the punchline. There are search clouds. These are really efficient search clouds. It's as if it's searching its environment randomly. We have tested this experiment where we give it a cue. Even if the cell is right there, it doesn't see it. It's like a graduate student that's hungry and is not essentially smelling the food and just walking around everywhere. But in this context of a tethered active filament, and this is exactly what we did. One of the things you can see is that some of the modes that encode this search behavior, there is activity in this system in which the thing that you saw in terms of turning on and off is essentially cilia on its head. So it's this filament that turns on and off activity at its tip. And that reversal is where the retraction is. Turning on is the activity in the front. You can see that it generates a lot of flow fields. One of the beautiful things about this, and it comes back to geometry, and I don't have time to talk about it, there is a microtubule machinery that encodes this shape. It cannot take any random shape space. There is actually a very constrained sets of shapes that it can take that are driven by the coupling between microtubules, membrane, volume conservation, and activity. I'm just going to tell you what activity does. Long story short, this is an object that's essentially a chaotic oscillator. What we have done here is the simplest possible model, a single static filament with activity at the tip that's turned on and off periodically, model all the elasticity, all the hydrodynamics in the system. And what we find is essentially this. For a broad range of parameters of activity and elasticity, this turns into a chaotic oscillator. You can try searching and finding cycles in this. And since we don't have any time, I'll just show you this. And we have proven at this point that this is actually a chaotic oscillator. And essentially, it searches space. One way of thinking about it is forget where you were as quickly as possible, and then you get a random search. Ironically, in certain sets of parameter spaces, it also has beautiful periodic dynamics. Some of the sets of, I'll let you watch this and you can try to guess for those sets of parameters what these cycles look like. We have both odd and even period cycles itself. And you can see right there, that's a 10-period cycle, 6-period cycle, 2-period cycle. But majority of the time, for the largest amount of phase space, this is actually a chaotic oscillator. Now, simply thinking about this idea is not new, but its instance in a biological context is new. You can go tonight home, another homework for you, turn your garden hose on at full pressure, but don't hold it at the tip. Hold it way further back. This is called the garden hose instability. Because the force that's being applied at the tip is what a kind called follower force, in which the force direction changes with the instabilities of the filament, you essentially get these sets of systems to behave completely differently from the classic Euler buckling types of frameworks. The real reason much of this framework was actually discovered was in rockets. People were trying to engineer rockets and finding sets of problems associated with rockets being unstable. Okay. Since I don't have any time, I'm just going to share puzzles and a quick sets of videos and let you ponder and read through some of the literature. Another puzzle that we've been thinking about is how do animals that don't have any neurons behave? It turns out there are two phylas. Neurons didn't appear out of nowhere. There was a significant portion of time in the animal kingdom where there might not have been anything like a neuron. And it turns out even today, there are actually two phyla, sponges you might know of, and the other one that we study are Placozoa, don't have any neurons. So how would they encode behavior? And one of the things that we think about in this context is, since this is going to turn it into a video talk, I just want to show you the framework for what these animals do. Now, this really, in my mind, is Flatland. I don't know how many of you read that book, Flatland. It's interesting in many ways, but it really inspired many people to think about mathematics and dimensions. This is a pancake animal. This animal lives itself in the bottom of the ocean. It's a deflated beach ball. It's only 20 microns high, but it can be as large as actually a few millimeters. These are all clonal animals, and I'll let you watch this, and you can see incredible amount of behavior. This animal is a clonal animal to this animal. They don't care about any shape and form, which is what we are told, that animals develop into specific shapes and forms. One of the things that we were able to discover in the system is actually an origin of a mechanical brain. Very much, and again, much of the origins of that behavior arises in a topological context of sets of defects. The cilia that are driving the system, and literally millions of cilia, interact and act as a ciliary flock that encodes a kind of a defect. That defect is essentially the memory in the system that encodes behavior. And so if you were to actually watch how the cell in cross-section is walking, is not a swimming organism, it's a walking organism, and you can see individual sets of cilia walking across, and the flocking dynamics encodes the kind of behavior. And I'm just going to show you, skipping over my hundreds of slides. In a live animal, what I'm highlighting here is just the defect now, a defect in the ciliary dynamics. And you can see these defects are incredibly complex. They can arise, they can be encoded from the boundaries, they can break, they can flare. And all of this sets of pseudo particle right here, which is really, there is no defect per se other than in the ciliary orientation. That is what's driving the real-time behavior of a system like this. And in the context of thinking about what that looks like at single cilia resolution, I'll let you watch this. We record millions of cilia simultaneously at single cilia resolution. I don't know how many of you can see. There's a little defect there that's propagating. These are some of the smallest animals. We have larger animals with much more complex number of defects. Okay, now maybe I should just tell you something about the ocean and probably end the talk and turn it into a discussion. I hinted towards toroidal cells. Let's talk a little bit about topology for a second. I've been wondering for many years now, why can't I find cells that are toroidal? That's a false start. I did find that cell on a foldscope in the Caltech pond. By every definition, it looks like a donut. Did anybody figure out the donut puzzle? Not yet. Okay, keep thinking about it. Because when donuts divide, some bizarre things happen. This is a false start. It actually turns out that is an amoeba. It's not donut-like, its shell is donut-like. Topology in that context will create all kinds of problems if the cells were toroidal. And we started thinking about this in the context of the ocean, trying to search cells that are deep in the ocean. This is much of the work is with two fantastic postdocs. Rahul is here and Adam Larson. We spent some time on a boat. We put in a little collector. We go down around between 100 meters, 200 meters, which is in the dark zone. We collect cells. And we found something that was quite surprising. We spent a lot of time on boats. And since I don't have time to tell you that, I just want to show you this toroidal cell that we have discovered. The puzzle associated with this cell is we find it in the dark zone, but it requires photosynthesis where there is no light. So how the hell? And of course, it's been labeled as non-motile. Nobody knows that it actually moves. So how the hell the cell, we can even find it all the way down to 200 meters. How the hell would this cell? And by the way, this happens to be pyrocystis. If any of you have seen bioluminescence, this is the cell that generates bioluminescence. When we look at the structure of the cell, we made a remarkable discovery. Again, going back to topology, It's an N genus Taurus. So I'm just going to let you watch this cell. Adam has been printing these cells. I'm happy to pass this around. This is a small enough crowd. This is the actual cell geometry. That's the cytoplasm. It doesn't exist in a giant ball. It exists in a reticulated network. So pass this around. And topology matters in this context because this cell does something incredibly bizarre. It expands like a balloon, the largest volume expansion that we have ever seen in a cell, around 10-fold volume expansion. And I'm just going to show you that volume expansion. Now you can think why topology is so important. So this is day-night imaging. The cell is just sitting there. It's moving its plastids around. And I know it's a video, so it will happen. At some point of time, you will see a dividing cell right there. You see that inflation? That was 5 minutes, and essentially a 10-fold volume increase. If you think about a cell in the cytoplasm in any biochemistry, that much dilution, it's taking water in. That much dilution should kill every enzymatic activity that you might see. But because of this topology, turns out it's linked with a vacuole. So the cellular network is only one-dimensional, while this three-dimensional expansion actually occurs in the vacuole itself. This is what enables it to balloon. You might ask, why would the cell want to balloon? Going back to Kilo Moana in Hawaii, the cells use ballooning to go up and down. Ballooning is actually motility. It's activity. Most of the time, cells that we have thought have been non-motile, that have no flagella, no cilia, are still motile. The moment it balloons, it takes a ton of water, which is lighter than seawater, and hence rises up. You can see the vegetative state rising. This cell can rise 200 meters. And one of the things, it's completely independent of orientation. It can be sloshed around and still make it to light. It needs nutrients to go down and light for photosynthesis. And the cell essentially has this incredible, bizarre lifestyle. Okay, I should end with an unpublished puzzle for you that you should try to solve yourself, driven by a video like this. So I'm not going to tell you how this works, but this is related to a geometrical property of spirals. You're all in a spiral right now. You may or may not realize it. Spirals can be found all the way at nanometer scale to galaxy scales. I've been interested in cyanobacteria for a while. There are kinds of fossils that are almost a billion year old, which can still have these spiral type structures. And the question that we've been interested in is, in the lab, we can create, naturally, systems can create these spirals. I'm just going to show you the picture of the spiral. Just let you stare at this for a second. It's a purely geometrical object. Literally, this is a puzzle that you can solve if you doodle around. What you're watching are individual cells. These are cyanobacteria, chains of bacteria, very long, that move both forward and backward. Every one of the cells is labeled. You can just watch the cell. It's going forward and backward. But it's trapped in the collection of these sets of cells, of these sets of objects that it will make. If you watch any filament long enough, there is flux along the radial axis. So it's not as if they're all completely trapped. And this dynamical structure is stable for days. One of the puzzles that we were interested in is, of course, what's going on. I don't have time to tell you that, but I'm going to show you something quite geometrical, which we are now calling topological traps in these systems. So first of all, we often understand these systems in the context of dislocations. You can think about this as a crystalline structure, because only topological changes in the system happen at these endpoints. So the very first model that you can write is purely geometrical. These are dislocations moving around. Any of you have taken any classic crystal mechanics will be able to do something like this. We can write down all possible rules in the system. We can simulate the system. But here is something that's very interesting. In some of the structures, we see flux. And in some of the structures, we see a flux barrier. Something about it fundamentally stops all flux to happen at that boundary point. That makes the structure stable. Now, you can doodle for hours and think about this. or I can give you the answer. Actually, maybe I'll stop here. If you ask me, I'll give you the answer. Otherwise, I'll let you ponder. Something purely geometrical in this system enables this property. It's a topological property of spirals. Spirals found around each other. Let me end here by saying something that's far more important than anything else that I have said, which is, we can all ponder and wonder, unless we give access for everybody else to engage in that process, we will not fulfill our belief of those tens of millions of species even being observed at the time that we are losing species faster than we can discover them. This has bothered me for almost a decade. In the old days, we used to be better. We used to invite friends on what were called microscopy clubs on breakfast and just sit down and ponder. Everyday people, sometimes people would look horrific looking at that their drinking water has monsters. This is the Victorian era. And unfortunately, we have lost that joy amongst everybody. Almost a decade ago, we started thinking about this problem that led to an origami microscope that some of you have seen. It's a $1.75 microscope. This allows people to see the cellular scale. The Upload's video I showed was actually taken by a kid in our community using a tool that costs less than $2. This is a very large community. In fact, it's actually the world's largest amateur microscopy community, roughly around 2 million people. These people may or may not look like scientists. They may or may not look like us every day. One of the contexts is we have the privilege that we have to share our curiosity and to explore our curiosity. It needs to be with everyone. These people might be fishermen. They might be rangers that are protecting our environments. They might be kids that have been in a war zone. It might be people who are much more excited about the microscopy world than their own kid. So they're not sharing the microscope with their child. They share every single day. 15 minutes before the talk, somebody in the community just asked me, oh, could you identify the cyanobacteria for me? Even before I could respond, somebody else from some other part of the world actually responded, oh, this happens to be a nostoc. It's a related species to the one that we study. This is a massive database now of millions of people walking around and collecting cellular scale data to explore the world that we all deeply care about. I'm going to end on this note. There's a lot of work that community publishes by themselves, but most importantly, it's important when we are thinking about science is what role does everybody as an individual play in science? Who defines what that would be? This is one of my favorite and most joyful and the most depressing picture that somebody in the community shared with me. I love this picture because you can see the kind of excitement. This is a rural village in Tamil Nadu. After English, Tamil is the most spoken language on our platform because of an individual that started training kids. 4 years later, he's trained around 100,000 kids in this community. The sense is that even a small little thing that we can do allows somebody access. We have reached, it's been 10 years. We've reached around 2 million people. There are 2 billion kids on this planet. Half of them, 1 billion or so, live by definition of the UNICEF under poverty lines. So one of the things when we think about every day, you get a chance to explore your curiosity. Remember the privilege that you have. Remember that if you give, you will get far more back than what you might realize. I'm going to end here. I hope you all have your own little creational biology and try to share that with people. Thank you. Far more difficult to give a talk at Aspen. I can't breathe.
Questions. Let's have a little conversation in the time we have left. Yes.
No, you said you would tell us how the organism creates a boundary.
Okay, I'll tell you.
Can you repeat the question?
Yeah, the question is, how does this spiral structure create a boundary? It actually took me an embarrassingly long period of time to figure this out, but I'm very delighted because nobody has written about this in the context of spirals. Something quite beautiful is happening here. When you look at a structure, any spiral, there is a movement in that spiral, which is the direction that it's moving, there is a geometrical chirality in the handedness that it was coiled. So if I just look at that red filament, you can see it's coiled counterclockwise. The arrow rotated counterclockwise. We call that geometrical chirality in this system. Suddenly, if I now take that purple filament, it's moving clockwise. The geometrical chirality for that would be clockwise. Now, if you mix these geometrical chiralities together and let the system move, whenever you have clockwise and a clockwise, and a counterclockwise and a counterclockwise, they can topologically intersperse. They can entangle. You can see the collisions no matter whichever direction they're going. At those collision points, they have the chance to go from the outside to the inside, because this filament has an opportunity now to either slide on top, slide inside. But when you do this exact same thing in the context of a counterclockwise and a clockwise, or a clockwise and a counterclockwise, geometrically, it's impossible in 2 dimensions. You've now entangled this in a topological state. There can be no inversion of these filaments, no matter how much they turn, whichever direction they turn. Then when you go back, look at this. This is a topological boundary right there. So any amount of flux that would happen at that point, you have created this mixture. Because new filaments come randomly, either in clockwise and counterclockwise, you eventually, as much material you have, the spiral keeps growing. Because every little bit of time, there will be one moment where you would get this cross chirality, the spiral is locked again. Spiral is locked again over periods of time. And one of the reasons I find this beautiful is this might be one of the reasons we don't have any proof for this, why multicellularity was incredibly important to make large-scale biofilms of these types. So effectively, this film is always starting to grow, no matter what these cells do, unless they jump over each other to be able to escape this trap.
Other questions? Yes, ma'am.
Could you explain or define for Toroid?
Yes, toroid. So
Question, Manu?
the question was, how do you define the word toroid? I think donut. By the way, did anybody figure out the donut answer?
10.
10. That's at least a good start. You could go a little higher. How many? 12. Not quite. Keep thinking. So a donut is essentially a toroid. And the simplest way of thinking about this is, because this is a topological property, that no matter how much you move around a donut, you cannot turn it into a sphere without breaking a boundary. And I'm trying to look for a nice picture for this, which is here. So you can think of a sphere, but once you have created a hole, you have created this topological property, which is invariant to any amount of shape change in this object. This object cannot be transformed into a sphere. That's so the toroid is the first genus when you have one hole. And what I meant by an n genus object is when it has n number of holes. And this cell, it turns out, the cell that I was talking about, is actually an n genus torus, in the sense that it has all of these holes. It's only this reticulated network that's connected together. And there is no way, without fusing and breaking all of these membranes, that you could actually turn that into a sphere. Then comes the problem of how the hell does this cell divide, which is what we are currently working on.
Final questions? Manu, do you want to tell them if they can play with stuff if they come up to you after?
Yeah, I have a foldscope right here. If you've never used it, we picked up some soil up at the gondola. We dug the snow until we got to soil. I promised Sujit I'll show him some soil, and you can come look at that soil right here, and then we can talk about it. And then again, if you want to just come and talk, I'm here.
Thank you all so much for coming. And just a reminder, the craft shuttle does pick up at the 15 and 45 at 5th and Gillespie. So in 15 minutes, there will be a shuttle back to town.